essentially JNK1 is bad and why we want to inhibit it. They're not really super biochemy, so if we want to take them out, we can. I don't know how much more biochem stuff I can add to them. So starting off, a 2-hybrid assay technique was done in rats and was used to find the site of interaction on IRS1 for JNK1. And they found that IRS1 has 2 LXL motifs between these residues that I have listed here, and that these 2 LXL motifs were found to bind specifically to JNK1. On what residues of JNK1, I have no clue, but this is what they found. Okay, so they did something called, like I said, a 2-hybrid assay. So in a 2-hybrid assay, we're looking for protein-protein interactions. So in our experiment, we're looking for the interaction between JNK1 and IRS1. And so we're going to look for this interaction in yeast. And what's going to happen is when JNK1 and IRS1 come together, we're going to get the activation of we're going to get transcription of a reporter gene. In this case, the reporter gene lacks SED, which in turn is going to promote beta-galactosidase activity, and we're going to get yeast growth on our Petri dish. So this is kind of full of animation, so hopefully it makes it easier. So again, here we see that we have, we have 2 specific domains. So here we have a binding domain, and we have an activating domain. So the binding domain and the activating domain are both needed in order to have transcription of the LAC-C gene to make yeast on our Petri dish. So this again is our lac operon, going back to grade 12 in first year bio. So what they did was kind of ingenious in my eyes. What they did is they took the activation, IRS1 was coupled with the activating domain, and JNK1 was coupled with the binding domain that was attached to the reporter gene. And so what happens is that when the activating domain and the binding domain come together, so when IRS1 and JNK1 come together, we're going to get transcription of the gene, and we're going to get yeast on our plate. So pretty cool. So again, they just tacked on those domains to the 2 proteins, and now we're going to see what happens. So here we see that JNK1 and IRS1 having its 2 LXL motifs, when they bind, transcription is going to occur. So we get the binding, we get transcription, we get our yeast. So what they did is they said, okay, that's cool. They hypothesized this LXL region. So in another 2-hybrid assay, they knocked out the 2 LXL motifs, and the experiment was repeated. And so IRS1 lacking these binding residues was not able to bind to JNK1, and therefore we didn't get any yeast growth. So you see again, so he's saying, oh no, not binding to JNK1, I can't because I don't have these LXL motifs, and again, we get no yeast on our plate. Very sad. Okay. No yeast. No yeast. So now we're going to look specifically at JNK1 and IRS1 interaction. So specifically serine 307 phosphorylation. In the notes I specify whenever serine 307 comes up that serine 307 is in rats, whereas serine 312 is in humans. So again, you'll see some experiments where they're talking about 307, others when they're talking about 312, and just realize that the difference is that 307 is in rats and 312 is in humans. So serine 307 is adjacent to the PTB domain of IRS1, so I feel like this is pretty clearly labeled for you guys. So the PTB domain interacts with the juxtamembrane region of the insulin receptor. This is going way back, but hopefully that jogs your memory a bit. And JNK1 is one of the kinases which phosphorylates serine 307, again in rats. So the 2 roles of serine 307 phosphorylation by JNK1 are to inhibit the interaction between IRS and the insulin receptor, as well as decrease insulin-stimulated tyrosine phosphorylation. So we see here that it has two main roles. The first one is going to be inhibiting them actually coming together, and the second role of serine 307 phosphorylation is going to inhibit tyrosine phosphorylation. So this should have an X through it eventually. So now we talk about some experiments, and this is where things get a little bit tricky. I feel like the notes I wrote were pretty well explained, so I'm just going to kind of read off the notes and show you where I'm talking about. First, this column here, the IKK alpha, is going to hopefully be eliminated. If someone has Photoshop or can do this, I have no clue how. So essentially, to determine whether excuse me, to determine whether JNK1 mediated phosphorylation of serine 307 in rats inhibits binding between the insulin receptor and IRS1. So again, this is one of the two roles of serine 307 phosphorylation. We had another yeast assay that was conducted. So again, this is just yeast growing on plates, nothing too exciting. So yeast cells expressing the human insulin receptor and human IRS1 constructs grew efficiently when inactive JNK1 was expressed. So in this chart, JNK1 APF is going to be inactive JNK1. I should probably add like a legend or something here explaining, just for your guys' sake, or maybe even blotting these out and adding new labels might be helpful. So again, when we had in yeast, when we had IRS1 with the insulin receptor in yeast, and we had inactivated JNK1, we had yeast expression. So this is really good. Activated JNK1, on the other hand, prevented growth, suggesting that the insulin-1 receptor interaction was inhibited. So that's right here. So again, they did essentially the same thing with a 2-hybrid assay technique. So they had, as we saw again before, how we had when IRS1 and JNK1 came together, we had the transcription of the reporter gene. So this is the exact same thing except with IRS1 and the insulin receptor. So when they come together, we're going to get transcription of the gene and yeast on plate. But this is essentially like a tri-hybrid assay in the sense that we're also adding either active or inactive JNK1 to the mix. So essentially what this top part of the experiment is showing us is that, EV is also a control well, just for all y'all want to know. But so essentially what this top part of the experiment is showing us is that when JNK1 is active and present, it's going to inhibit the binding of IRS1 to the insulin receptor. So now we're moving down here to this well. And so I have that the substitution of serine 3